Hi, this is Duncan Ferguson. In this unit, we're going to talk about the metabolism of thyroid hormones. In particular, we'll focus on the deiodinative metabolism. And forgive me if I get excited about this because I did my PhD in this subject. So, some big picture things about deiodination. I wanted to start with that and the metabolism of thyroid hormone. Most tissues are capable of deiodinating thyroid hormone. There are a few that have low activities, but we know there are now 3 different deiodinase enzymes. When I did my PhD many years ago, we only thought there was one that converted T4 to T3 in tissues like the liver and the kidney. What's most important is to recognize that this is sort of a local tissue regulation of the amount of thyroid hormone activity that tissue wants and needs, if you want to put it that way, at any given time. And that allows each tissue to sort of, almost like a thermostat, to turn that room up or down based upon its metabolic functions. And then, sort of to be complete, we need to think about conjugating mechanisms for thyroid hormone elimination. Usually these are glucuronides or sulfates, and these are the conjugates that are eliminated through the bile, the liver and the bile, or via the kidney. And so they're important to us because they do have an impact on the overall elimination of the hormone, but also in the case of dogs and cats, they have a very efficient way of wasting, if you will, excess amounts of thyroid hormone that we might give them as clinicians or in an oral route, by an oral route. So let's talk about the other types of metabolism pathways, the conjugation first. And first of all, it's interesting to note that there's huge amounts of T4 and T3 that exist in the gut at any given time in the form of these conjugates. This conjugation mechanism by the liver is a major mechanism that prevents animals from having an accidental overdose. And we can give sometimes bottles of T4, or an dog could eat a bottle of T4 and not really be impacted by it as much as a human would be, because the mechanisms for elimination are so dramatic in the dog and cat. And one way to think about it, I like to think about it, these are carnivores, and the carnivores sometimes eat the thyroid gland, and so they may have developed mechanisms whereby this is a protective mechanism to keep a tiger, for example, from walking around the wilderness in a hyperthyroid state. We also know that thyroid hormone can be conjugated, and then once it gets to the colon, unconjugated again and reabsorbed. These conjugating mechanisms can be stimulated by drugs that stimulate the cytochrome P450 system and other elimination pathways, such as phenobarbital, the anti-epileptic drug. And so we have to pay attention to this when we look at animals that are on these drugs, because their T4 and even their free T4 levels can fall a little bit even when they're euthyroid. Sulfation is probably a more important pathway by which thyroid hormone can be lost into the urine through the kidney. So for this unit, this particular graphic is really important. We're looking at T4, first of all, here, and you remember that T4 has 4 iodines. There's 1, 2, 3, and 4. And we label those, and this is crucial for us to understand the names of these enzymes, as outer ring or inner ring relative to this amino acid side chain. And so there are 3', 5', or 3 or 5 sites for iodine. Let's deal with, going left, the activating forms of the enzyme are 5' deiodinase. So we call these are basically 3' and 5' are equal sites. So when we say 5', that could be either one of those iodines being plucked off and turning into T3, and we will learn The T3 is more active than T4, so this is considered to be an activating step. So there are two types of deiodinase, 5' deiodinase enzymes. One is deiodinase 1, and the other one is deiodinase 2, and we'll show you in the next graphic where these are located in the body. But let's, for the moment, just say they take T4 and convert it to T3. Then we have the inner ring deiodinases, or 5 deiodinases, and these, while deiodinase 1 has some activity along this line as well, I want you to focus on deiodinase 3. Deiodinase 3 was one of these enzymes that we didn't pay a lot of attention to because it just inactivated thyroid hormone. But we now know that in many places, such as the central nervous system and the placenta, that this enzyme is really crucial for inactivating hormone and not allowing it to be active until the appropriate time of development. But the product of this pathway from T4 is what we call reverse T3. So you can see this iodine has been removed, and that's why we call it reverse T3. For all intents and purposes, this particular metabolite has no activity. So you basically have an activating step going to T3, and that's the deiodinase 1 and 2, and then you have an inactivating possibility with deiodinase 3. As you go strip more iodines off of these molecules, most of these don't have much metabolic activity, although some of them are starting to be discovered as having some interesting activities, but quite different than T3 and T4. The main role is to take and recover this micronutrient iodide and allow it to be reused by the body. So here I want to focus a little bit about where these deiodinases exist. Deiodinase 1, and this is what I did my work on my PhD, is I did work in the kidney, and it's also in the liver. And I want you to think about this type of deiodinase being associated with making T3 and making it again available to the circulation. So these are highly perfused tissues that allow T3 to be released to other organs. Deiodinase 2, which we see in the brain, and we already mentioned the pituitary, and also brown fat being a more metabolically active form of fat, are the T3 that's made by these tissues and the iodinases inside the cell is actually retained in those organs, and so this is truly an enzyme that creates intracellular T3. Deiodinase 3 is, as I said, an inner ring deiodinase, and it's an inactivating 5 deiodinase, and it's seen in the placenta and in the brain, particularly during development. All of these enzymes are selenoenzymes, and so that means selenium is an important micronutrient. There are animals with selenium deficiency that can have thyroid hormone abnormalities. The other thing I wanted to explain is that when we say outer ring or inner ring deiodination, we can remove iodine. You can see from the pathway on previous slides that it doesn't make any difference whether you're taking an iodine off T4 to make it T3 or reverse T3 to make it 3,3' T2. These are iodines being stripped off that are then recovered by the body. And the same thing with inner ring deiodination. So the pathways are parallel on either side, inner ring, outer ring. And as we said, beyond T3 and reverse T3, deiodination continues to recover iodide for the body. Here I have a graphic showing the location relative to a cell, and this is a sort of prototypical cell. Wherever these deiodinases exist, you can see that deiodinase 1 is a plasma membrane, has a plasma membrane location. So does deiodinase 3. And so think about deiodinase 1 as kind of doing its thing, releasing T3 to the circulation, and deiodinase 3 doing the same thing. It's sort of protecting that cell from thyroid hormone activity by taking T4 or even T3 and inactivating it. T3 and T4 can get into cells by transporters, and we won't talk about that now, but there are specific transporters, and depending on the tissues, for these hormones to get into the cell. Otherwise, they can enter by diffusion, as shown here in the bottom right. Within the cell, we have deiodinase 2, and I mentioned that it's mainly converting T4 to T3, and that T3 becomes intracellular T3 that then can move to the nucleus and have its action. So, again, deiodinase 1 and deiodinase 3 sort of impact circulating plasma T3 or reverse T3, and deiodinase 2 seems to be providing in tissues where it is in high quantity intracellular T3. In this graphic, I want to let me show you how the deiodinases 1 and 2, or what we call type 1 and type 2, 5' deiodinases, kind of regulate in different ways when the body is put under certain circumstances. We're going to focus on fasting and hypothyroidism. But let me talk you through this. So, in the circulation, we have plasma binding proteins. We have T3 and T4 in equilibrium in the free forms here. And let's focus on free T4 right now. In our sort of rectangle here on the right, let's say this tissue is the liver. So it's a deiodinase 1 tissue, and you can see that deiodinase 1 there. You know now that it really would sit on the outside of the cell, but let's just think about it. So this tissue, this free T4, comes to the tissue, gets deiodinated, T3 is made. There are cytosolic binding proteins that kind of pick it up inside the cell to keep thyroid hormone happy, and then this T3 acts on the cell, and it can do things like change metabolism, alter protein synthesis, oxygen consumption, etc. In the oval, we have, let's say, the brain or the central nervous system, and here we have deiodinase 2, and it converts T4 to T3 and does the same thing. So why am I showing you all this? What is different about these two tissues, if we think about them as the liver and the brain, is when the animal is fasted. The liver, deiodinase 1 activity, so fasting, and we'll go over this in the next slide, makes deiodinase 1 go down. Fasting. And in the brain, deiodinase 2, no change. Why would this make sense? Well, the liver is a metabolic organ, thyroid hormone is a metabolic hormone, and when the animal doesn't have nutrients, remember we said there's this big matching thing going on between nutrients and oxygen consumption or metabolism. That's why this mechanism occurs. In addition, let's now talk about hypothyroidism. So when an animal becomes hypothyroid, and that means there's very little thyroid hormone available to tissues, so that basically, let's say free T4 goes down, and it goes down, deiodinase 1, so we look at hypothyroidism, deiodinase 1 goes down, okay? In hypothyroidism, deiodinase 2 goes up. Why would it go up? Well, it's the body saying, okay, in hypothyroidism, I'm lower metabolism, I don't need as much stuff done by the liver, it goes down in the liver. The deiodinase in the brain says, no, I can't stand being hypothyroid, I want to protect myself, and so when I am hypothyroid, this deiodinase 2, and this is where that negative post-translational feedback comes in, it actually makes deiodinase 2 hang around longer, and T3 levels go up higher, so the intracellular, the amount of T4 converted to T3 percentage-wise increases, and therefore that's protecting the brain from the hypothyroid condition. So you can see that these two different types of tissues with two different types of deiodinases have two different responses in these changes of metabolic state or nutritional state. So to summarize some of these changes, basically anything that associated with what we call protein-calorie malnutrition will lead to reduced T3 production, particularly in the liver. In addition, in situations where we have chronic illness or what we call non-thyroidal illness, meaning that it's not associated with some problem with the thyroid directly, sometimes called NTI, this will also lead to a reduction of T3. And why would this happen? Well, it's believed that the reduction of T3 happens to protect the body from not wasting too much amino acids, breaking down of protein in the muscle, leading to gluconeogenesis. So this is a sort of a buffering effect to keep from overt amounts of protein catabolism. We also see effects of stress, and glucocorticoids can also have effects on some of these pathways. The important thing for the clinician is that these changes are physiological or pathophysiological. They're not pathological, meaning that hypothyroidism is not what the body is thinking. The pituitary doesn't think it's hypothyroid, it thinks it's euthyroid. And so TSH tends to be normal. It will only fall very late in disease and with very severe illnesses, and this is probably another mechanism. So let's look at some of these. We already mentioned fasting as being affected with a reduction of T3 and a rise of reverse T3 when deiodinase 1 will go down and deiodinase 3 may go up. Exercise in an individual or an animal or person that has balanced energy intake will have no change in the system. That's sort of supportive of the idea that if the caloric intake and the energy use is equal to each other, you're okay. But if you don't have an adequate amount of intake, like a marathoner not loading with carbohydrates ahead of time, then during that race, their T3 may fall. Similar changes have been seen in dogs that do the Iditarod, for example. On the other side, overfeeding or obesity, while the changes are variable, we sometimes can see increased free T4 in the cat. That's probably due to free fatty acids going up. But you also can see an increase in T3 and reverse T3. And there is some evidence to suggest there is a little bit of a moderate hyperthyroidism in this situation. So in general, the regulation can go both ways. We already mentioned illness, and generally we see reduced T3 and increased reverse T3 in human, dog, and cat, and it takes very severe illness before we start to get a suppression of T4, and that's probably due to pituitary TSH secretion falling eventually in severe illness. So in summary, deiodinative metabolism is the most important form of thyroid hormone metabolism, as it is regulated by nutrient availability and illness. The deiodinase 1 is an activating 5' deiodinase. It's in high quantities in the liver and kidney, and it tends to be the type of deiodinase that determines circulating T3 concentrations under euthyroid conditions. The most important thing there is that it helps to deliver activated thyroid hormone T3 to other tissues. Deiodinase 2 tends to be intracellular, and it's in the nervous system, and the T3 is generally made and retained locally. Deiodinase 3 is an inactivating or deactivating inner-ring deiodinase, 5-deiodinase, that's more expressed during development and then can be increased during malnutrition and illness that leads to the elevation of reverse T3. And in some tissues, circulating T3 concentrations may not be very helpful to know what's going on in those tissues because they're making their own T3 from T4. And so we should think about T3, therefore, as an intracellular hormone and T4 mostly as an extracellular hormone. And probably the T4 concentrations, therefore, are more reflective of the animal's thyroid status from the standpoint of how much the thyroid is secreting.